All right, so we would like to start with welcome message. This workshop is organized by Bioimaging Facility and UBC Dynamic Brain Circuit Cluster and UBC NeuroImaging and Neurocomputation Centre. And we're just gonna introduce ourselves and start off with myself. I'm Miki Fujita. I'm a research manager in UBC Bioimaging Facility. And we have Yoon. Say hello, Yoon. Okay, so Yoon is our optical microscopy staff in UBC Bioimaging Facility. And Jeff.
And my name is Jeffrey Ledoux. I'm a staff person at the Center for Brain Health, and I'm working with the NeuroImaging and Neurocomputation Centre and the Dynamic Brain Circuits Cluster. And we're also joined by Keegan Flanagan, who is a summer co-op student with the Brain Circuits Cluster.
All right, and just we want to spend some advertisement, some time to advertise our facilities in case you don't know us. And let's start with Jeff's facility.
Okay, I will share my screen. So just a quick overview of these shared resources at the Center for Brain Health. So I'm responsible for this NeuroImaging and Neurocomputation Centre. It's a core facility that focuses on preclinical imaging and microscopy, as well as computation and data analysis. And then INC functions in combination with another group called the Dynamic Brain Circuits Cluster, which promotes collaboration and open science practices. These are distinct entities, but very overlapping. The NINC is mostly about the equipment, and the Brain Circuits Cluster is mostly about people. Just a quick overview of what we have in the NINC. So there are 7 imaging systems. These are largely confocal and 2-photon laser scanning microscopes. We have a data analysis and computation lab with GPU-enabled workstations, a small compute cluster, equipment for tissue clearing and expansion microscopy, and a makerspace where people can fabricate parts for their experiments. We did have some success in the last round of Innovation Fund with CFI, so we're going to expand this facility and incorporate some new technologies like lattice light sheet microscopy, fiber photometry, 3-photon microscopy, ultra wide field of view 2-photon microscopy, ultrasound imaging, and add 700 cores and 2.5 petabytes of storage to Compute Canada. So that's coming in the next few years. With that, maybe I'll turn it back over to you, Mickey, for the BIF.
I'm just gonna spend 2 minutes or 1 minute to talk about our facility. So UBC Bioimaging Facility is located in a biological science building, basement of the North Wing. And if you're interested in our facility, we have this website, bioimaging.ubc.ca. And if you have any questions about facility, please send me an email. So our facility is open to everyone, and before COVID time, before pandemic, we had about 200 researchers coming from different research disciplines every year. And we provide training and service, and we have 3 full-time staff who can provide you training and service. And we have range of optical to electron microscopy, and we have also sample prep equipment, imaging, and image analysis capabilities. And just quick overview of what we have. Optical microscopes, we have a regular conventional light fluorescence confocal microscope. We can do live cell imaging, multiphoton imaging. If you want to do high resolution imaging to nanometer level, you can do electron microscope. We have SEM, EDX, TEM, and also 3D TEM, correlative light electron microscopy. And we are getting new equipment which can do 3D imaging using SEM. So we can use focused ion beam to mill surface and imaging, mill away, imaging and mill away, so 3D imaging. And then we have sample preparation equipment, including cryo fixation and chemical fixation, mainly for electron microscopes. All right, so I'm stop sharing. Are we doing okay with time? All right. So, it's about time to start the first session. So I just gonna screen share. Actually, I can go through today's schedule. So we start with 45 minutes lecture talking about guidelines for appropriate use and manipulation of scientific digital images. And after that, we have 10 minutes break. And then we have 45 minutes hands-on session. We try Fiji software, how to do the basic image processing. And we have 10 minutes break. And the last session is ImageJ macro. So, okay. So before going to first lecture, I want to talk about something about research misconduct in image processing. So most of the microscopy images, it comes with digital format. It's easy to manipulate, and sometimes those images considered as research misconduct. And what is research misconduct? U.S. Department of Health and Human Services Office of Research Integrity defines misconduct as fabrication, falsification, or plagiarism in proposing, performing, or reviewing research, or in reporting research results. And research misconduct does not include honest error or differences of opinion. And research misconduct is a very serious issue because it affects research integrity and impacts reproducibility of the outcome. A lot of people want to follow materials and methods described in the paper, and they want to reproduce the data. Then if the images are inappropriately manipulated, then it questions is it really results that they got, and then sometimes it results in paper retraction. Also, it can damage reputation and career. So if you have chance, I recommend to visit this ORI website. It's shown in the bottom side of the slide. And also slide will be available after this workshop. So when it comes to digital image, what kind of digital image related misconducts are there? So there are two types of manipulations considered as misconduct. First one is inappropriate manipulation, which refers to adjustment of image data that violates the established guideline, but does not affect the interpretation of the data. So the question is what those established guidelines are. So we're gonna cover this in the first session. The second type of manipulation is fraudulent manipulation, which refers to adjustment of image data that does affect the interpretation of the data. So it's really intentional manipulation of the image, which is not allowed to do for scientific purpose. So I just want to ask you this quiz. I want you to think about which one is considered as misconduct. Select all that apply. I'm not going to give you answer. I'm going to provide answers in the end of first session. So let's see. First one, A, brightness contrast adjustments that fades or vanishes part of the image. Is this considered as misconduct or not? B, cleaning up the background. Is it proper to do this? C, enhancing a specific feature with the intent to support the proposed hypothesis. D, cropping that changes the context of the final image. E, cloning an object into an image in which it did not appear previously. F, color adjustment for readers afflicted by color blindness. So I'm gonna give a minute to for you to think about. Great, so I'm gonna move on. So I will start with the first lecture, 12 guidelines for the appropriate use and manipulation of scientific digital images. So I'm gonna be presenting this lecture and Yoon is gonna be available in the chat area. So if you have any questions, please put them in chat and you will answer questions. And if you have time in the end of this session, I will try to answer all the questions you may have. So objectives of today's first half portion of the workshop is to understand what is a digital image, identify what image data manipulations are considered acceptable, and then the next session, edit images using basic tools available in ImageJ Fiji. And my first take-home message is that images are more than just pretty pictures. So microscopy-based experiments can provide you with both qualitative, pretty pictures, and quantitative, which has meaningful numbers, and you can do quantitative analysis and statistics. So it's scientific data. And first of all, treat images as data. So digital images are composed of pixels and each with specific location relative to its neighbors and the scale relative to the instrument used to capture the image. And each pixel is assigned a numerical value, intensity, pixel intensity we call it. So here we have grayscale image, and if we zoom in, region of interest, and we can see each pixel, and those pixels compose of this area of the image. And each pixel has those numbers Those ones are indicating intensity of each pixels. And if you look at this top right corner, black pixel, the pixel intensity, the numerical value is 0. And if you look at this white pixel, this pixel has pixel intensity 255. And if you look at pixels with gray color, they have the numbers between 0 to 255. And if you quickly scan all the numbers, and you will find the lowest number is 0 and highest number is 255. There's a reason, because this image is 8-bit grayscale image, which has a variation of 256 intensities from 0 to 255. So when we have digital image, the digital image has two important informations. The first one is the number of pixels used to create the image. We call it pixel resolution. And this pixel resolution is totally dependent on the pixel number on the chip in a camera or detection system you use. So most of the microscopy system, it has 1K camera, which means the camera can provide 1024 pixels by 1024 pixels. Some cameras are 4K camera, means that the camera can generate 4048 pixel by 4048 pixel image. And second important information is the number of colors. We call it color depth or bit depth, measured in bits. For grayscale image, it's shade variations. So for example, if you have 8-bit grayscale, the image has 256 variations, color variations, shade variations. So 2 to the 8th variations. If you have 16-bit grayscale, then 2 to the 16th variations, which means more than 65,000 variations in a pixel intensity. And then if you have color camera attached to the microscope, then those color camera can generate color image. Then let's say you have 8-bit RGB image. RGB stands for red, green, blue. Then what it means is that 256 intensities for each red, green, blue channels. Then the image has more than 16 million color variations. So if you can have high bit image like 32-bit, 24-bit images, you have more color variations in an image. That's great, but then the size of the image file increases, and some of the image analysis tools doesn't work in high-bit images, and sometimes we have to convert it to 8-bit to do image analysis. So we have to tweak around this bit size, but most of them the image analysis processing tools can handle 16-bit images. Number 2, save the original, work on the copy. So it's really important you save the original image and you don't touch those original image. When I say original image, it contains both raw image plus metadata. And raw image is, so I have the example of image here on the left-hand side, and right-hand side is a screenshot of metadata. And I hope you can read some of the descriptions here. In the metadata, we can see like when the image was acquired, which microscope system was used, which camera was used, And also it has pixel information. So here you can see 1024. This is pixel number. Also here in the image size, it says 1024 pixels about this image. It's equal to image size about 159 microns. And this image is 12 bits. And also we can see objective lens information. And if you're using confocal microscope, it also shows up laser intensity, gain setting, and pinhole size, and blah, blah, blah. So it's very important to keep those raw data separately from other files. And any image manipulation is done on a copy of the original. And once you do something with the raw image, please change the file name and save as. Sometimes when you are submitting the paper in a journal, and reviewers ask, please submit original image and original raw data. If you are not able to do that, then your results are not considered as real results. Like you have to be able to present both raw image all the time and also edited image and well documented what you did to the image. Number 3, making simple adjustments. Reasonable linear adjustments to brightness and contrast are appropriate. You can do that. There are other adjustment. It's called gamma adjustment. This is non-linear adjustment setting, and this is questionable. If you have to apply this gamma setting, you have to document in materials and methods of figure legends. And all changes must be documented. Avoid to use automatic image adjustment. Sometimes the software has auto function. If you hit the auto, your software does something or usually applies complicated algorithm. You don't know what's going on. And you want to make it simple when you do adjustment of the image brightness and contrast, for example, and you want to make it under your control. So avoid to use automatic function for the image adjustment, and always examine the histogram. So the histogram shows the distribution of the numerical values of all the pixels. So here I have an example image of plant seedling. And here you can see the pixel information, how many pixels are in this image. And this is 8-bit grayscale, and this is the size of the file. When I look at histogram of this image, it shows the distribution of the pixel intensity. So x-axis is pixel intensity from 0 to 255 because this image is 8-bit. And y-axis is count of pixels. And here it shows that this image has about 8 million pixels in the image, and mean value of pixel intensity is about 210, and standard deviation is around 19. And minimal pixel intensity is 92. So we don't have complete black pixels in this image, for example. So the pixels intensity starts from 92 to 254. So let's say I want to do brightness and contrast adjustment. So the goal of image adjustment is to stretch and even out the histogram. So here is again this image of the plant seedling, and this is the histogram of this image. And here is the same, it's the same histogram as above on the left-hand side. And when I do linear adjustment, I use this minimum and maximum bar to cover the pixels that have pixel intensity from 92 to 254. So here in the example, I'm selecting the pixels from that have pixel intensity 91 to 255. And if I apply this linear adjustment, well, when I do this linear adjustment, I don't choose from, for example, somewhere around 150 because I'm eliminating or removing the pixels that have less than 150 pixel intensities, and you can not lose any pixel information. So make sure that you're covering all the pixels from minimum pixel intensity to maximum pixel intensity when you do linear adjustment. Then here is the after adjustment. So you can see the histogram. If you look at and compare the histogram, the original histogram is stretched out and image looks somewhat having more contrast. And both images are acceptable for publication, but just a matter of which one you think it's proper to, like, it depends what you want to show in a video. So that's brightness and contrast adjustment. We also gonna cover this in the next session using Fiji software. Okay, so number 4, process the entire image evenly. So you should never use the dodge and burn tools in Photoshop. And dodge and burn tools are heavily used in the photography field. And if you're using Photoshop, you can see those functions, but please don't use those functions. And what those tools do is they selectively apply less or more light to defined areas and extremely questionable practices. So I have the example image before and after. And there's cherry blossoms in the center of the image. And dodge and burn tools, you can lighten up all the flowers and then kind of darken all the background. And those tools are not acceptable when you're dealing with scientific digital images. Number 5, use of software filters to improve image quality is not recommended. Simply put, avoid to use filters. There are some filters like smoothing edges or like kind of make your images beautiful. We call it data beautification. Those ones are not acceptable. And there are a lot of filters available in Photoshop, but please don't apply those filters. An exception, some cases when you do image analysis, you have to segment your research of the region of interest. You may need to use those filters. We are hoping to cover them in a future workshop, but the principal rules, please avoid to use filters. Number 6. Cloning or copying objects into a digital image is very questionable. Use of the clone tool in Photoshop, please don't do that. You can add additional information. You can remove some information you don't want. That's considered as intentional image manipulation. Sometimes it is used to clean up an area of the image, clean up the background, but then you may be removing or adding data to other part of the image. And it's better to avoid doing that. And if you have to use this cloning tool, must be documented in the figure legend or materials and methods in the paper. Number 7, cropping an image is usually okay, but if cropping changes the context of the final image, it is not okay. So I have an example image. This is an insect of SEM image, original image at the top. And here I have, well, all the SEM image comes with the information about scale bar and image parameters. And those, for example, this bottom part of the image is not necessary when I want to put this image on for the publication. And I'm interested in head region of this insect. Then it's okay to crop this area to show region of interest. And here is my final image for publication. So I crop it and I added scale bar in the image. Number 8, comparing images. So when comparing images, if it's whether qualitative or quantitative, images should be acquired under identical conditions. So when you plan your experiments and conduct experiments, please make sure that you're using the same setting, image acquisition setting. And you cannot compare images captured under different conditions. And examine the metadata to determine conditions. And some microscopy system, the software has kind of reload your setting, image acquisition setting, so you don't have to manually enter all the parameters, but use the same setting for all the images. Also, the image processing editing tools you apply should be identical to all the images when you compare images. Number 9, making intensity measurements. If you're dealing with fluorescence microscopy images, you may want to do intensity measurement. And generally, it is very challenging to do appropriately. Image acquisition parameters and post-processing techniques need to be standardized. When you are doing imaging, well, there's a laser fluctuation happening, and sometimes your fluorescence signals start to photo bleaching, fading out. And there are some hardware optical aberrations of you have to prepare sample, same for each experimental setup, and must be performed on uniformly processed image data when you make intensity measurement. And we recommend to have positive and negative controls along with your experimental setting. And robust, repeatable standard is required. And often the data must be calibrated to a known standard such as fluorescent beads. And if you're using you're using the fluorescence dyes, and you can use anti-fading reagent in mounting media to prevent photo bleaching. And number 10, insert a scale bar on the image. So magnification is determined by the difference between the original scale and the scale in its final form. And if we have image and it says, oh, this image was taken by 60 times objective lens, it doesn't give us any information about scale of this image. And please make sure you insert scale bar in each image when you're presenting images. It's very important. And we're gonna cover this in the next session, how to insert the scale bar using Fiji. Number 11, avoid the use of lossy compression. Save images in TIFF format. So there are two types of image file compression. First one is lossless file compression, TIFF. So TIFF reduces the size of the file, but retaining the integrity of the data. So it retains the pixel resolution, number of pixels in the image, and also color depth, 8-bit, 16-bit. If you save your image as JPEG or PNG, then you lose all the pixel resolution and color depth, bit depth. And you lose all the resolution. So do not save your images in JPEG, PNG. Just like, yeah, those compressions, JPEG compression algorithm is performed, and then, yeah, you lose all the resolution. Even you use very high-end microscope and very high resolution camera. So yeah, I just wanted to point out. And some people use PowerPoint to make figure panels, but once you import image, insert image in a PowerPoint file, the default resolution is screen resolution 72 dpi. So you are losing resolution of the image. So it's not recommended to use PowerPoint file to prepare figure panel for publication. Number 12, so be careful when changing the size in pixels of a digital image. And here I'm showing the screenshot of Photoshop. So let's say you're preparing image for publication. Most journals require 300 ppi, point per inch, or dpi, dot per inch images. And we have this raw image. Just in a Photoshop, if you go to image size, you can see all the information about pixel resolution. So this image has 973 pixels by 975 pixels. The image size is 10 inches. And if you look at the resolution, this image has 96 ppi, which doesn't meet requirements for the journal. And I want to convert this image to 300 ppi resolution. So as a default, oh, by the way, Photoshop is available for all the research labs in UBC. So yeah, if you don't have one in your laptop, please use your lab computer to use Photoshop. So this Photoshop default has those two check marks. And just let's say if you're okay with this default setting, then what you get is, okay, resolution becomes 300 pixels per inch. That's great. But you can see the pixels, number of pixels increased. This is not acceptable. You cannot add pixels. You cannot remove pixels from the image. And this is not acceptable to do this. Okay, so how we can do this conversion? So when you convert the image from 96 to 300, just check the last bottom, resample image. Please deselect this option and press OK. Then the pixel resolution stays same. The image pixel numbers are 973 by 975. And then you may notice the size of the image decreased from 10 inches to 3 inches. And resolution is 300 pixels. So that's the correct way to convert the size in pixels of a digital image. All right, so other things. So take-home image messages are to use ImageJ Fiji to process digital images, and use Photoshop to organize figure panels or change sizes, and never use PowerPoint to create image panels for publications. It's okay for oral presentation, but when it comes to manuscript, preparation of manuscript, please don't use PowerPoint. And this lecture series was initially provided by previous manager of our facility, Garnet Martens. He started to educate our users what we can do or what we cannot do for the image editing. And I put link to ORI, and also there's a paper talking about these guidelines. When you have chance, I recommend you to visit this paper. And yeah, I have 6 minutes. So which one is considered as misconduct? A, brightness contrast adjustments that fades or vanishes part of the image. It's really hard to say. It depends on the situation, but we want to avoid that happen. And sometimes it considered as misconduct. Cleaning up the background. If you are using cloning tool, maybe you didn't do intentionally, but it can be considered as misconduct. So please be careful doing that when you do that, and please document when you do something on the image. Enhancing a specific feature with the intent to support the proposed hypothesis. This is intentional manipulation of the image. It will consider as misconduct. Cropping that changes the context of the final image. Yeah, if, for example, you have gel image, like there are several bands, and then here is additional band you don't want to show. You crop the image, then yeah, you cannot do that. So cropping that changes the context of the final image. That may be considered as misconduct, how you do that. And cloning an object into an image in which it did not appear previously. That's right, it's considered as misconduct. F, color adjustment for readers afflicted by color blindness. It's not misconduct. Usually the default setting of the image software gives you red and green combination. And often we recommend to convert them to cyan magenta combination, just for color blindness. Yeah, it's better to show or convert from green-red combination to cyan magenta. Okay, so I also want to introduce this 7-minute YouTube video, which is really interesting, that how we can detect those manipulations that we talked about, like inappropriate image manipulations. And this is 7 minutes. So if you have time during the break, yeah, you can watch it. But also I want to remind you during the break, we ask all users to open Fiji on your computer and have BIF sample images available on the desktop. And breakout room is available for those who need assistance. And we're gonna put links in the chat. I'm just gonna stop sharing.